Hey everyone, Mohammed Hamama here. Welcome back to the ASCP preparation camp. This is our second video in the microbiology module, and today we're diving deep into bacterial cell structure. We're going to peer right into the blueprint of bacteria, arguably the most successful life forms on Earth. We're not just talking about single-celled organisms. We're breaking down the unique architectural decisions they've made, that cell envelope that dictates exactly how they operate, and more importantly, how we can target them clinically. It all starts with a foundational difference. All bacteria are prokaryotes, meaning they're structurally much simpler than our cells. They lack a defined nucleus, and their DNA is typically just a single double-stranded molecule. But here's the critical thing. While their internal metabolism might have some similarities to ours, the structures they build, especially that outer shell, are entirely unique. And those unique structures are the exact points we have to target if we want to defeat them. So let's zoom right in on that bacterial cell envelope, this sophisticated interface between the cell and the outside world. It's composed of the cytoplasmic membrane, the cell wall, and then in some cases, an outer membrane and a capsule. This envelope really defines the cell's vulnerability. Starting with the innermost layer, the cytoplasmic membrane. It's more than just skin, it's the absolute line of demarcation, the cell's most restrictive boundary. Like our own cells, it's a phospholipid bilayer. You've got the polar phosphate groups, the water-loving parts, facing outward, and the nonpolar lipid tails all tucked inside. Its primary job is to be a highly selective permeability barrier, rigorously controlling what comes in and what goes out. But if it's so restrictive, that raises a really interesting question about logistics. How does the bacterium get the huge complex building blocks it needs for the cell wall, which is outside this membrane, across it? That's a fundamental biological puzzle, and the answer is specialized carrier systems, which we'll get to later when we talk about antibiotics. Outside that gatekeeper membrane is the structure that defines all bacteria, the cell wall, built almost entirely of peptidoglycan, or PG. Peptidoglycan is truly unique to prokaryotes. If you see it, you know you're looking at bacteria. What makes this structure so incredibly strong? It's essentially a cross-linked meshwork with immense tensile strength. You can break it down into two parts. The glycan part is the carbohydrate backbone, these long linear sugar strands alternating between two subunits, N-acetylglucosamine, or NAG, and N-acetylmuramic acid, NAM. That gives you the vertical strength, like the long rungs of a sugar ladder. The peptide part provides the horizontal scaffolding. These are short amino acid chains that cross-link adjacent sugar strands together right at the NAM subunits, creating a really tight, stable network. What's fascinating is this evolutionary defense that's built right into the structure. These peptide chains often incorporate D-amino acids, like D-alanine. Now that seems like a tiny detail, but it's a brilliant defense mechanism. Our own biology, the enzymes we produce to break down proteins, they're almost all specialized to recognize L-amino acids precisely. By using D-amino acids, the bacteria build a wall that is inherently resistant to being broken down by the peptidases we might use against them, like the ones in our tears. It's like walking up to a lock and finding the keyhole is reversed. That structural choice, how the envelope is built, brings us to probably the single most critical distinction in microbiology: gram-positive versus gram-negative. This separation, which comes from the gram-stain procedure, is entirely based on cell envelope architecture. Gram-positive organisms keep it simple but very robust. A thick, multilayered peptidoglycan wall sitting right outside the single cytoplasmic membrane. And because that wall is so thick, it holds onto the crystal violet stain used in the process. What are the defining markers on that thick surface? They feature something called teichoic acids, polymers of substituted glycerol units covalently linked right into the PG structure. They essentially poke out from the surface, so they're the first thing our immune system sees. They're a major cell surface antigen. Gram-positive bacteria also have lipoteichoic acids, which actually span the whole PG layer and anchor right into the membrane itself. If gram-positive is defined by one thick wall, gram-negative is all about complexity and layers. They have two membranes, an inner cytoplasmic membrane and a distinct outer membrane. Their peptidoglycan layer is extremely thin and it's sandwiched between the two membranes, housed in a space called the periplasmic space. That periplasmic space is a whole functional compartment. Not only does it hold that thin PG layer, which makes gram-negatives mechanically a bit weaker, but it's also packed with enzymes and transport proteins. This dual membrane system means they have an extra barrier that many antibiotics have to cross. The real structural challenge, the component that makes gram-negative bacteria so uniquely dangerous, is embedded in that outer membrane, lipopolysaccharide, or LPS. It's the defining feature of the gram-negative outer leaflet. It has 3 regions, but the most clinically relevant parts are the O polysaccharide, which is highly variable and used for strain identification, and lipid A. Lipid A is the toxic part. It's an integral part of the membrane itself. When a gram-negative cell is just growing, it's fine. But when it dies, when the cell gets lysed by the immune system or, ironically, by antibiotics, lipid A is released into the bloodstream. That's why it's called an endotoxin. It causes systemic inflammation, fever, and if the bacterial load is high enough, it can lead to lethal septic shock. Just to be clear, it's an endotoxin because it is a part of the organism itself, released when it dies. We differentiate it from exotoxins, which are proteins that are actively secreted by the living cell, think botulinum toxin. And then we have enterotoxins, which is just a functional class. They're exotoxins that specifically act on the gut. The big takeaway for gram-negatives is that killing the cell releases a potent poison. Tricky targets indeed. External appendages. Moving beyond the cell envelope itself, bacteria have these external appendages for moving around and getting a solid foothold. Let's talk motility, that means flagella. These are long helical structures made of a protein called flagellin, and they're fascinating molecular machines. The engine is the basal body, a ring-like structure anchored in the cell membranes that rotates the whole filament like a screw propeller. It's often called one of the only true rotary motors in nature. Just stunning nanotechnology. What does it let them do? It enables directed movement, or chemotaxis. Lets them swim toward good things like nutrients and away from bad things. And of course, because they're so large and prominent, they are highly antigenic, easily spotted by the immune system. Then we have the smaller appendages, the pili, sometimes called fimbriae. Pili are much shorter and thinner than flagella, and their main job is adherence. They're not for moving, they're for sticking. They're microscopic grappling hooks that function as attachment structures promoting specific cell-to-cell contact. Bacteria use them to attach to specific host cells, like the ones lining the urinary tract, or even between one bacterial cell and another. Some specialized pili are critical for transferring genetic material in a process called conjugation, which is a key way antibiotic resistance spreads. That final protective layer, the sticky coat, or glycocalyx. This is a viscous layer of usually polysaccharide that's secreted outside the cell wall. We distinguish it based on how organized it is. If it's tightly bound, it's a capsule. If it's loose and amorphous, it's a slime layer or glycocalyx. What are the capsule's survival powers in a host? It provides multiple layers of protection. First, adherence, allowing cells to adhere to surfaces. Second, and this is critical, protection against phagocytosis. Immune cells just can't grab onto encapsulated bacteria as effectively. They really struggle. Third, it acts as a physical diffusion barrier, offering some protection against antibodies and even some antibiotics, thus contributing to the organism's pathogenicity. It also protects against desiccation, drying out, which really helps in airborne transmission. One fascinating exception, Bacillus anthracis, the cause of anthrax. Its capsule isn't polysaccharide, it's made of polydiglutamic acid. A great example of chemical variation for survival. Antigenic variation. Because the immune system is always looking for these surface structures, LPS, capsules, lipoteichoic acids, pili, and flagella, bacteria evolved a counter-strategy, antigenic variation. This is the expression of various alternative forms of antigen on the cell surface, and most surface structures are subject to it. This variation is important for immune evasion by the pathogen. For example, in Neisseria species, antigenic variation by gene conversion allows the organism to produce antigenically different pili molecules at high frequency. So it's constantly changing its surface markers to stay one step ahead of our antibody response. It's a constant chase. Variation in these surface structures between strains of the same species can be detected by serology. Bacterial spores. If a bacterium faces a truly existential threat, extreme heat, nutritional deprivation, environmental hostility, some of them, mostly gram-positive rods like Clostridium and Bacillus, have the ultimate survival mechanism, endospores. These can be released from the original cell as free spores, and they are the ultimate survival pods. They're not for reproduction, they're metabolic survival machines. The cell undergoes profound structural and metabolic changes, essentially repackaging a copy of bacterial DNA into a new form that contains very little water, has no metabolic activity, does not divide, and has a restructured, highly impermeable, multilayered envelope. What gives them this unbelievable resistance? Spore formation begins with the invagination of the parent cell membrane, producing a double membrane, the forespore, that encapsulates and isolates a copy of the bacterial DNA in what will become the core of the spore. The process then involves synthesizing this incredibly thick, impermeable, multilayered envelope. Inside, the mature spore retains the complete machinery for protein synthesis, and new spore-specific enzymes are synthesized in the core. The core has very high levels of a unique compound called calcium dipicolinate, which is thought to be essential in stabilizing and protecting the spore DNA from heat and chemical damage. Many enzymes of the original vegetative cell are degraded. When the endospore is completed, the parent cell lyses, releasing the spore. The result is unparalleled resistance. Spores are the most resistant life forms known. They survive boiling for hours, intense UV, and standard chemical disinfectants. This is why sterilization procedures like autoclaving have to be validated by their ability to kill spores. Spore germination. To return to the vegetative state, spores must first be subjected to a treatment that weakens the spore coat, such as heat or extremes of pH, thus allowing germination to occur. This is called activation. If the activated spore is in a nutritious environment, which it senses by monitoring various key metabolites, it begins to germinate. This process involves destruction of the cortex by lytic enzymes, followed by uptake of water and release of calcium dipicolinate from the cell. Which brings us to the classic food safety example, Clostridium botulinum spores. It's a perfect illustration. Some of the most notorious pathogens are spore formers, including Bacillus anthracis, which causes anthrax, Bacillus cereus, which causes gastroenteritis, Clostridium tetani, which causes tetanus, Clostridium botulinum, which causes botulism, Clostridium perfringens, which causes gas gangrene, and Clostridium difficile. These spores are common in soil, so they get on vegetables. You take a baked potato, wrap it in foil. The temperature inside the potato during baking stays below 120 degrees Celsius. Not hot enough to kill the spore. Not even close. And the foil creates an anaerobic environment. So when that potato is stored at room temperature, any surviving spores can germinate, and the vegetative cells then produce the lethal botulinum toxin. The risk is so high because killing the active cell or the toxin only takes 100 degrees Celsius, but killing that spore requires temperatures above 120 degrees Celsius under elevated pressure, that you only get from an autoclave. The spore survives everything but the autoclave. In the absence of an autoclave, spores can be largely eliminated by a primary boiling to activate germination, and after a short period of vegetative growth, a second boiling to kill the now vulnerable vegetative cells. Let's shift now to the life cycle. If bacterial cells are suspended in a liquid nutrient medium, they follow those four famous phases of growth: lag, log, or exponential, stationary, and death. And since they divide by binary fission, 1 becomes 2, 2 become 4, 4 become 8, the number of cells increases exponentially with time. That speed of growth is just astronomical. A species like E. coli can divide every 20 minutes depending on the medium and conditions. You start with 1 cell, and in 8 hours you have around 10 million cells. The log phase, the exponential phase, is the point of fastest growth. It's probably the most critical period in an infection. Why? Because the cell's metabolism is at its maximum. It's constantly building large structures, especially that peptidoglycan cell wall, getting ready to divide. If we hit them with drugs that target those synthesis pathways, they're maximally vulnerable. Eventually, growth slows and ceases entirely in the stationary phase as nutrients are depleted and toxic waste products accumulate. Most cells in stationary phase are not dead, however. If they are diluted into fresh growth medium, exponential growth will resume after a lag phase. The stationary phase presents different challenges because bacteria often form protective biofilms that shield them from drugs. We mentioned metabolic flexibility earlier. How do they generate all the energy for this rapid growth? A distinctive feature of bacterial metabolism is the variety of mechanisms used to generate energy from carbon sources. They show far greater variety than we do. We classify it into 3 primary types. First, aerobic respiration, where oxygen is the final terminal electron acceptor of the electron transport chain, like us. In this process, oxygen is reduced to water. This is the energy-generating mode used by all aerobic bacteria. Second, anaerobic respiration, where inorganic compounds other than molecular oxygen serve as the terminal electron acceptors. Depending on the species, acceptors can be molecules like nitrate or sulfate. Anaerobic respiration can be used as an alternative to aerobic respiration in some species, facultative organisms, but is obligatory in other species, some obligate anaerobes. And then there's fermentation. Fermentation is an anaerobic process where an organic metabolic intermediate derived from a fermentable substrate serves as the final electron acceptor. The substrates that can be fermented and the final end products depend on the species. By comparison to aerobic and anaerobic respiration, fermentation yields very little energy. The purpose of fermentation is not really energy creation, it's critical waste management. It's all about recycling the coenzyme NADH back to NAD. That ensures glycolysis can continue and the cell doesn't choke on its own metabolic intermediates. The terminal electron acceptor in fermentation is pyruvate or a pyruvate derivative. These end products can be measured and are sometimes diagnostic for a given species. Which brings us back full circle to our ultimate antimicrobial strategy: disrupting that cell wall synthesis. It's a brilliant target because it's a structure our cells just don't have. If we look at how the PG wall is built, the bacterial peptidoglycan polymer is constructed on the surface of the cell membrane via a series of distinct stages we can target. Stage 1 is preparing and activating the building blocks inside the cytoplasm. As in all biological polymerizations, NAM and NAG subunits are activated by attachment to a carrier molecule, in this case, the nucleotide uridine diphosphate, or UDP. A pentapeptide is then added to UDP-NAM by sequential transfer of amino acids, and that's where we find that diala-diala dipeptide, which is critical for cross-linking. The two terminal alanine residues are added as this dipeptide. The antibiotic cycloserine inhibits the synthesis of that unique diala-diala dipeptide block, the foundational piece. You stop the dipeptide, you stop the whole wall. Stage 2. Getting that completed NAM peptide unit across the membrane. The NAM pep moiety is transferred from the UDP carrier to another carrier, bactopenol phosphate, or BPP, located on the inner surface of the cell membrane. At this point, UDP-NAG transfers NAG to NAM pep, completing the peptidoglycan repeat unit, NAG-NAM pep, which is now attached to the carrier BPP. BPP then carries this repeat unit through the cell membrane to the outside surface. After delivery, BPP has to be recycled to carry the next unit, and that's where bacitracin works. It inhibits the recycling of the BPP carrier. If the carrier can't be recycled, transportation shuts down. The final critical step, stage 3, addition of the repeat unit to existing peptidoglycan and cross-linking outside the membrane. This is where the heavy hitters operate. BPP carries the NAG-NAM pep repeat unit to the outer surface of the membrane where the peptidoglycan of the existing cell wall is located. The repeat unit is added to a free end of the existing peptidoglycan, increasing the length of the polymer. This stage is governed by two major drug families. First, vancomycin. It works by physically binding to the terminal diala-diala dipeptide after it's been transported but before it can be incorporated into the growing peptidoglycan chain. So it just gets in the way, acts as a massive physical obstruction, blocking the enzyme from getting to the site. A pure steric blockade. Because vancomycin binds to the terminal diala-diala dipeptide, this antibacterial agent also prevents transpeptidation. And then the most famous group, the beta-lactams, penicillins and cephalosporins. Beta-lactams are unique. They block the actual cross-linking reaction itself. Although the N-terminal end of the pentapeptide is attached to the NAM moieties of the backbone, the C-terminal end is dangling free. Cross-linking is brought about by a transpeptidation reaction. Beta-lactams inhibit the enzymes that catalyze these transpeptidation and carboxypeptidation reactions of cell wall assembly. These enzymes are called penicillin-binding proteins, or PBPs, because they all have active sites that bind beta-lactam antibiotics. No single PBP species is the target, rather, their lethal effect on bacteria is the result of inactivating multiple species of PBPs. Most PBPs are involved in bacterial cell wall synthesis. Inactivating PBPs is usually lethal, but this is the constant battleground. Acquired resistance to beta-lactam antibiotics often happens when bacteria evolve genetic modifications that result in production of new PBPs that have lower affinity for beta-lactam antibiotics, lowering the drug's binding and letting that vital cross-linking continue. That wraps up our discussion on bacterial cell structure. Make sure you understand the differences between gram-positive and gram-negative envelopes, the stages of cell wall synthesis, and how different antibiotics target each stage. These concepts are absolutely critical for the ASCP exam. In our next video, we'll be diving into bacterial genetics, looking at how bacteria transfer resistance genes, plasmids, transposons, and the mechanisms of conjugation, transformation, and transduction. You won't want to miss it because genetics ties directly into everything we've covered today about antibiotic resistance. If you found this video helpful, please hit that like button, subscribe to the channel, and turn on notifications so you don't miss any videos in our ASCP Prep series. Drop any questions in the comments below. I read them all and I'm here to help you succeed. This is Mohammed Hamama, and I'll see you in the next video. Keep studying, stay focused, and let's ace that ASCP exam together.